Okay, so hello everyone. Thanks so much for coming in person. My name is Dena. I'm a fourth year PhD student in the Liebel lab, and today I'll be talking about my project looking at the role of glial D-Serine in synaptic damage and cognitive dysfunction after brain injury. So traumatic brain injury, or TBI, can occur through a wide variety of mechanisms, including a direct mechanical impact to the head, rapid acceleration and deceleration, or blast waves. And traumatic brain injury is very prevalent. It affects nearly 70 million people worldwide each year, resulting in over 2.5 million visits to the emergency room, almost 300,000 hospitalizations, and over 50,000 deaths in the US alone each year. And TBI results in a wide variety of symptoms, as you can see illustrated here. This includes things such as motor impairments, behavioral changes, such as increased risk for depression and anxiety, as well as cognitive impairments. So when patients are admitted to the hospital following a severe TBI, the standard of care is really to promote survival past the first few weeks of injury. And this can be done by categorizing and monitoring patients based on various factors, such as intracranial pressure, cerebral perfusion, and brain oxygenation levels, where the standard of care is really aimed at maintaining homeostasis of the brain. And there's really no standard therapies to minimize damage from secondary injury mechanisms beyond just promoting survival. So that's something that our lab is interested in, is to better understand the molecular mechanisms of injury to provide more targeted therapies for neuroprotection. So when we talk about these molecular mechanisms of injury, this can be split into two phases, the primary injury and the secondary injury phase, where the primary injury occurs at the time of impact. And this includes things such as direct damage to blood vessels, blood-brain barrier damage, as well as tissue and axonal shearing. So besides preventing the injury itself, there is really not much that can be done to therapeutically treat primary injury. So we're really focused on these secondary injury mechanisms, which occur in the minutes, days, or even months and years after the primary impact. And this includes things such as ischemia and edema, excitotoxicity due to excessive glutamate release, mitochondrial damage and oxidative stress. And what I'll be focusing on in my project is synaptic damage and gliosis. So before I get into synaptic damage in the context of injury, just wanted to go over normal synaptic functioning. Whereas we're all familiar with, NMDAR receptors are the primary excitatory neurotransmitter in the brain. And these are unique in that in addition to the binding of glutamate, they require the binding of a co-agonist, which in the case of the forebrain is D-serine. So when these conditions are met, this leads to calcium influx into the cell, which can lead to changes in gene expression and long-term potentiation. However, in the case of injury, too much calcium can also lead to excitotoxicity and cellular damage. So our lab is particularly interested in studying the role of D-serine, where D-serine is physiologically synthesized from L-serine by the enzyme serine racemase, which is expressed predominantly in postsynaptic neurons. And D-serine is then released via amino acid transporters, where it binds to NMDAR receptors at synaptic sites, leading to normal synaptic transmission. And under physiological conditions, glial cells, such as astrocytes, play a homeostatic role, where they uptake extracellular D-serine. However, what we've noticed in our injury model is that reactive glial cells will upregulate serine racemase expression, which leads to our overarching hypothesis that the hyperactivation of NMDAR receptors by glial D-serine causes synaptic and cognitive damage after injury. So in order to study synaptic damage, our lab uses a controlled cortical impact model, or CCI model. And in this model, a highly pressurized piston will deliver an impact over the intact dura. And as you can see here, compared to a sham image on top, there's severe cortical tissue loss in this model. However, the underlying hippocampus remains intact. And when we took a closer look at this, we saw that there were no significant changes in the number of new and positive cells in the hippocampus after injury, as shown in black for the CCI mice. However, when we quantified the number of synapses, we did see a significant decrease in synapses after injury, suggesting this is an advantageous model to study synaptic damage without cell loss. And so we wanted to see whether these synaptic deficits resulted in functional changes. So to do this, we use the fear conditioning assay, and this is a contextual-based learning assay where mice are placed in an environment where they receive an auditory cue followed by an aversive stimulus, such as a foot shock. And then 24 hours later, they're placed back inside the same environment where we measure the percent freezing. So a larger percent freezing corresponds to greater ability to associate the environment with the aversive stimulus the day before, indicating more learning and memory. And what we see here is that after CCI injury, we have a reduction in freezing based on contextual learning when the mouse is placed back in the same environment, as well as cue-based learning when the mouse hears the auditory stimulus, suggesting that our model does induce deficits in learning and memory. So as I said, we're interested in studying the role of D-Serine in these learning and memory deficits. So we wanted to look at D-Serine expression levels after injury. And for this, we can use serine racemase, the enzyme that synthesizes D-Serine, as a proxy, where serine racemase expression corresponds to D-Serine expression. So we can see here in on the left in sham conditions that serine racemase is localized predominantly in the neuronal layer of the hippocampus. However, 3 days after injury, there's a more diffuse expression pattern, which suggests a more glial expression pattern. And we also see enhanced expression of GFE positive astrocytes and IBO1 positive microglia cells after injury. And when we took a closer look, we do see colocalization of serine racemase with both the astrocyte cells and microglia cells after injury. And we also quantified this using flow cytometry, which isn't shown here, but we do see a significant increase in the percentage of both cell types that express serine racemase after injury. So to begin looking at the role of serine racemase in these specific cell types after injury, we have 3 different genetic knockout lines where we have the gene encoding serine racemase knocked out under the GFAP promoter, which is an astrocyte-specific promoter. And then we also have 2 different microglial mouse lines, where serine racemase is knocked out under the TMEM119 and CX3-CR1 promoter. So this would lead to a lack of D-serine synthesis in either astrocytes or microglia cells. And so we first wanted to see whether there would be any functional recovery by ablating serine racemase in either cell type. So to do this, we used our fear conditioning assay. And this is showing data from control mice at 7 days post-injury, where there is significantly less freezing in these CCI mice shown here. And when we looked at fear conditioning in both microglial mutant lines, we saw a rescue of fear conditioning behavior. And we also saw similar results when serine racemase was knocked out, specifically in astrocyte cells, suggesting that glial D-serine produced by either cell type is contributing to learning and memory deficits. We also wanted to take a more translational approach. So instead of genetically modified mice, here we used pharmacological inhibitors, where we delivered 2 different inhibitors, either L4 or BMS, after injury. And these are inhibitors that target D-serine transporters. So rather than preventing the synthesis of D-serine, here we're preventing its release. And these transporters are expressed predominantly on glial cells, and they're expressed on both microglia and astrocyte cells. So to deliver these, we use osmotic mini pumps. So these pumps can be implanted subcutaneously and are connected to the lateral ventricle using a cannula. So the mouse receives an infusion of the inhibitor for the 7 days after injury. And so we wanted to look at fear conditioning again in these mice. And here we see that control mice treated with a vehicle do have a deficit in freezing behavior. And this was rescued using both inhibitors that we tried. And this was at 7 days post-injury. So we also wanted to see if there was more long-term neuroprotection from this approach. So we looked at 28 days post-injury. And here we see that there is still the deficit persisting in the vehicle-treated mice after injury, which was rescued in the inhibitor-treated mice, suggesting there is long-term neuroprotection from inhibiting glial D-serine. And I'm not showing the data here, but we've also tested this using a genetic approach where we knocked out the SLC1A4 transporter for D-serine in both astrocytes and microglia cells. And we do see the same rescue as with the pharmacological inhibitor. So seeing these functional deficits, we wanted to see whether synaptic damage is underlying these deficits. So in order to do this, we use Golgi staining. So this is a silver-based dye that stains the entirety of the neuron, including individual dendritic spines. So this will allow us to count the synaptic density in the hippocampus, as well as look at the morphology of synapses. Where we're interested in dendritic spines that have this mushroom-shaped morphology, which is has a larger surface area and can receive more synaptic inputs. And this is indicative of mature synapses. We're also interested in thin spines, which are associated with degenerating spines, which we would expect to see upregulated after injury. So we first wanted to see whether these morphologies did correlate to learning and memory in our model. So here we plotted the percent freezing on the x-axis and the percentage of mushroom-shaped spines on the y-axis, and we do see a positive correlation where more mushroom-shaped spines correspond to more freezing, so more learning and memory. And we saw the inverse effect when we plotted the percentage of thin spines, where more thin spines corresponded to less learning and memory. So seeing that we do have this association between our Golgi data and our functional data, we then wanted to look at changes in our mutant mice. So first, looking at the controls, we saw significant decreases in spine density at 7 days post-injury. And we also saw significant decreases in the mature mushroom-shaped spines and significant increases in the thin degenerating spines. And when we looked at this in both microglial knockout lines, we saw a rescue of both spine density and morphology. And we also saw a similar effect with the astrocyte cells, suggesting that D-Serine from either microglia or astrocytes is sufficient to rescue synaptic damage. We also repeated this with our pharmacological inhibitor, where at 7 days, we still see a deficit in these spines in the synaptic density with the vehicle-treated mice, as well as deficits in morphology. And we saw that both of these were rescued with inhibitor treatment. And we saw the same effect at 28 days post-injury, where we still see decreased spine density in the vehicle-treated mice that was rescued in the inhibitor-treated mice. And we also did see the same results using our genetic approach, where we knocked out the SLC1A4 transporter in either cell type. So to look at synapses on a more functional level, the lab has also previously done electrophysiology. So these are long-term potentiation recordings performed in vivo in the hippocampus. And you can see here in black, these are the control CCI mice. So there was a significant deficit in long-term potentiation 7 days post-injury. But when we looked at the astrocyte knockout mice, we see no changes between these sham and CCI mice, suggesting that astrocytic D-Serine results in impairments in long-term potentiation after injury. The lab also performed a study where they infused D-Serine for 7 days after injury, and once again looked at LTP in the astrocyte knockout mice. So they saw that in red, the mice that were treated with vehicle still had preserved LTP after injury. However, the ones that had the D-Serine infused had a lack of LTP. And this is really supportive that it is D-Serine from these astrocyte cells that is contributing to deficits in LTP. And so moving forward, we do want to repeat this or conduct this as well in the microglial knockout mice to see if we see the same effect. Okay, so to summarize everything so far, in naive physiological conditions, D-Serine is synthesized from L-serine by the enzyme serine racemase, which is expressed predominantly in the postsynaptic neuron, where it's released via amino acid transporters to facilitate normal synaptic functioning. However, in our injury model, we see a downregulation of serine racemase in neurons and an upregulation in both microglia and astrocyte cells, where we found that either genetic ablation of serine racemase in either cell type or genetic ablation of the SLC1A4 transporter, as well as pharmacological inhibition of these transporters, were sufficient to rescue synaptic damage. And so we think that both cell types together are contributing to a threshold level of D-Serine, which leads to synaptic damage in our model. So this leads us to hypothesis that I'm currently looking at with some of my ongoing work. So this is what is the mechanism underlying glial D-Serine mediated synaptic damage following TBI. And so moving forward, I'll be focusing on the microglia cells, as this is really a novel cell type that we've first shown here is able to produce D-Serine. So we wanted to look more into the mechanism of how this is causing damage. So to begin looking into this, I wanted to look at differences in NMDA receptor signaling after injury with and without microglial D-Serine. So the two main subunits that we're interested in are the GluN2A subunit of NMDA receptors. So this is expressed predominantly at synaptic sites, and it has been shown to enhance the production of BDNF, which enhances learning and memory, as well as increase the phosphorylation of transcription factors ERK and CREB, which also lead to transcription of genes involved in synaptic plasticity, as well as cell survival. We're also interested in the GluN2B subunit, which has been shown in many models of disease, such as Huntington's, Alzheimer's, and ischemia, to inhibit some of these signaling pathways, leading to synaptic degeneration. So we first wanted to look at whether there were changes in these subunits after injury in our model. And we saw a significant upregulation of both the GluN2A and the GluN2B subunit at 1 day post-injury in control mice. And when we looked at this in the mutant mice, we saw no significant changes after injury, suggesting that microglial D-Serine is mediating the upregulation of NMDA receptor subunits post-injury. We also wanted to look at phosphorylation of the GluN2B subunit, as phosphorylated GluN2B has been shown to have increased conductance as well as stability in the membrane. When we first looked at the ratio of phosphorylated GluN2B to GluN2B, we didn't see any significant changes after injury. However, looking at total at phosphorylated GluN2B to total protein levels, we did see a significant increase in the control mice at 1 day post-injury that was delayed until 3 days in the mutants. So this is suggesting that while the ratio of phosphorylated GluN2B is not increased in our model, the enhanced expression of GluN2B that we're seeing is also likely phosphorylated, so meaning it could have more activity. We also looked at levels of DAPK, which is a kinase that leads to the phosphorylation of GluN2B, and we saw a significant upregulation of DAPK at 1 day in the control mice after injury, and this was delayed until 3 days in the microglial mutant mice. So this is corresponding to the time points we see with phosphorylated GluN2B, and is suggesting that this delayed phosphorylation could serve as a protective mechanism. We also looked at expression of other factors, including BDNF, which we saw was increased at an earlier time point in our mutant mice, which could lead to more synaptic plasticity. And we also saw increased phosphorylated CREB in the mutant mice and not the control mice. And as this is a transcription factor that can lead to transcription of many factors involved in learning and memory, this could be another form of neuroprotection. We were also interested in phosphorylation of ERK, another transcription factor. However, here we saw the opposite effects of what we expected, where we saw increased phosphorylated ERK at 1 day in the control, but not the mutant mice. And this could be due to the fact that we're looking at whole tissue expression here, where phosphorylated ERK has also been shown to be activated in many glial cell types under inflammatory conditions. So moving forward, we do want to conduct this on a cell-specific level. We wanted to look further into the GluN2B subunit as well. So to do this, we use pharmacological inhibitor romaliate, and this is specific to the GluN2B subunit over the GluN2A subunit. And here we use osmotic mini pumps to deliver the inhibitor for 3 days after injury, and then looked at analysis of dendritic spine morphology. So we see in black is illustrated mice that were treated with a saline vehicle, and we do see a significant decrease in the percentage of mature mushroom-shaped spines and a significant increase in the percentage of thin spines. And when these mice were treated with all 3 concentrations of the inhibitor, we saw a rescue of both the morphology of both the mature and degenerating spines, suggesting that inhibiting GluN2B can rescue dendritic spine morphology. So moving forward with this, we want to look at this using a genetic approach. So I'm currently working with GRIN2B knockout mice, and these are conditionally knocked out under the CAMK2 promoter. And we're looking at whether this GLUN2B subunit knockout has effects on fear conditioning, electrophysiology, as well as Golgi analysis. So just to summarize some of the clinical implication of this, here we identify that microglial D-Serine is a novel mediator of NMDAR receptor signaling after injury. And this is important as it provides a novel mechanism to modulate NMDAR receptors without affecting all excitatory neurotransmission. So rather than simply using antagonists for NMDAR receptors, we could target glial cells to kind of modulate this level of D-Serine. So moving forward, now that we see changes in NMDAR receptor signaling in our model, I wanted to look at whether downstream pathways are affected. For example, whether synapses are tagged with different proteins to identify them for synaptic pruning after injury, and if this is different in the absence of D-Serine. So before I get into this data, just wanted to orient everyone to the area of the hippocampus that I'll be looking at and some of the methods I'll be using. So we're interested in the CA1 region, particularly in the strata radiatum and molecular layer. And these receive a wide variety of different inputs, including from the entorhinal cortex and the C3 Schaefer collaterals. And this here is an image from a previous grad student in the lab. This is using tissue clearing, so we can see the neurons in green and astrocyte staining in red, where we see a lot of synaptic contact with astrocytes. And so we're interested in this layer here, the strata radiatum and the molecular layer. So using this, I was looking at the colocalization, or I will be looking at colocalization of different markers with pre and post synaptic markers in the CA1. So for this, I use staining with V-GLUT, a presynaptic marker, and PSD95 as a postsynaptic marker. And here, this is just to show what the strata radiatum looks like in the hippocampus and the molecular layer. And when we zoom into this, we can see colocalization of these factors in white. So first, I just wanted to see whether we were seeing similar results using this method as with our Golgi analysis, where we do see a trend towards less synapses at 7 days post-injury in the mutant mice. And this is very preliminary, but so far it seems like this is not the case in the mutant mice either. And so here we see about a 30% reduction in synapses at 7 days. And this is similar to what we're seeing with our Golgi analysis. So suggesting that this method is producing similar results with our injury model. And so since we're seeing some losses of synapses at 7 days, I wanted to look at various markers at an earlier time point before these synapses are eliminated. So moving forward, I'll be looking at the 3-day time point. So one of the first factors we're interested in looking at is phosphatidylserine. So phosphatidylserine, or PS, is a phospholipid that is normally localized to the inner leaflet of the plasma membrane. However, under conditions of cell stress, such as increased calcium entry, which in our case could occur through hyperactivation of NMDAR receptors, this leads to the externalization of PS, which then serves as a signal for phagocytosis. And this has commonly been studied in apoptotic cells, where the externalized phosphatidylserine serves as an apoptotic signal. However, it's also been shown to be the case in dendritic compartments, where exposed phosphatidylserine, specifically on dendrites and synapses, can lead to the elimination of synapses without cell loss. So we wanted to see whether this phosphatidylserine is externalized in our injury model and whether this is different in the absence of D-serine. So to do this, we used osmotic mini pumps to deliver a dye called PSVU. And this is because there are currently no good antibodies for immunohistochemistry of PS. And so we used osmotic mini pumps to deliver the PSVU dye for 3 days after injury. You can see here, this is an example of a sham tissue where we see virtually no PS presence. However, in the CCI, we see a lot of immunoreactivity in green in the cortex. But what we're interested in is the underlying hippocampus, which is outside of this main area of damage. So looking at this from a higher magnification, you can see individual puncta of PS, where here in a sham condition, we see very little PS puncta, and we see an increase at 3 days post-injury, which seems to be more in the control mice as opposed to the mutant mice. And so I looked at the colocalization of PS with the pre and post-synaptic markers, VGLU and PSC95. And this is a preliminary quantification, but as of now, it looks like there is enhanced PS colocalization with synapses after injury in the molecular layer, which is attenuated in the mutant mice. And there's more variability in the strata radiatum, but it does look like there is an increase, at least in the control mice at 3 days. So moving forward, I'm going to be adding more animals to this and see if there is in fact a difference between PS externalization with and without glial D-Serine. Another component I wanted to look at next was complement component C1q. So this is a component of the complement system that can tag synapses for elimination. And recently, complement component C1q has been shown to interact with externalized PS and is involved in pruning of synapses. And this has been studied in development. So we wanted to see whether this was true in our injury model as well. So we took the same approach, where here we see very little C1q puncta in either sham tissue, and we see increases in 1 day in the control mice here, as well as 3 days, and less C1q expression in the mutant mice after injury. And quantifying this, I did see significant increases in C1q at 3 days post-injury in the mutant mice, but in the control mice, but not the mutant mice, suggesting that microglial D-Serine contributes to C1q colocalization with synapses after injury. And next, we wanted to look at a novel receptor, GPR56, which has recently been shown to be able to interact with both phosphatidylserine and C1q and is involved in pruning synapses during development. So we wanted to take a look at GPR56 expression in our injury model, where here we used flow cytometry and we saw very little expression of GPR56 in sham conditions. However, we saw a significant increase in the percentage of microglia cells that express GPR56 after injury. And this was upregulated in both injury conditions. However, it was significantly less upregulated in the mutant mice, suggesting that interactions, there could be a positive feedback loop between interactions from externalized phosphatidylserine and C1q, leading to more expression of this GPR56. We also looked at the mean fluorescent intensity of GPR56, and here we didn't see any significant differences, but there does seem to be a trend towards more fluorescent intensity per cell in the control mice after injury. We also wanted to look at expression in astrocyte cells, but we saw very little expression in either the sham or injury conditions, suggesting this is a microglial specific pruning mechanism. So moving forward with this, I want to continue looking at the colocalization of phosphatidylserine with synapses, as well as C1q and C3, which is another member of the complement component implicated in synaptic pruning. And we've also generated GPR56 knockdown mice, where we want to see if a lack of GPR56 in microglia will lead to improved fair conditioning and Golgi analysis. So to summarize this section here, the clinical significance here is that this shows a novel mechanism for synaptic loss after injury that could lead to new avenues to protect cognitive function after TBI, such as, for example, targeting GPR56 to prevent synaptic loss. Okay, so next, I just wanted to note that everything that I've shown so far has been performed in male mice. However, I was also interested in looking at sex differences in our model because there have been many studies that have shown sex differences in injury responses. So here I first started just using our serine racemase knockout mice, the TMM119 microglial knockout mice. And I wanted to look at whether we see similar rescue using Golgi as what we did with our males. However, here we saw no significant deficits in the spine density or morphology in the female mice, even in the control mice after injury. And then we also looked at fair conditioning. However, we again didn't see a significant deficit at 7 days post-injury, as we do with the male mice. So since this was done in our mutant mice, these were all tamoxifen treated. And so we wanted to make sure that these effects that we're seeing weren't due to any neuroprotection from the tamoxifen itself. So I repeated this using wild type mice, where we see the same trend in fair conditioning behavior. So at 7 days, we have our learning deficit in male mice, but not in female mice. And we also wanted to look at this at a later time point. So I performed this at 6 weeks post-injury, and we still see that this deficit is persisting in the male mice, but not the female mice. So looking further into this, I'm currently looking at differences in NMDAR receptor signaling proteins in female mice after injury. And some of our preliminary results suggest that there aren't these upregulations in proteins in the female mice after injury. I'm also using the Morris-Water maze as a secondary assessment of learning and memory. And we're also planning on doing some single cell sequencing of hippocampal cells to compare differences between males and females after injury on a single cell level. Okay, so finally, we wanted to see whether these aspects of D-Serine mediated damage that we see in our mouse model are conserved in human TBI patients as well. So for this, we have a collaboration with neurosurgeons at Jackson Hospital, where during neurotrauma surgery, they will provide us with tissue that is acutely resected from patients. So this is looking at acutely injured tissue. So since first, we wanted to see whether everything that we've seen so far in our mouse hippocampus was conserved in the cortex as well, since many of the patient samples we receive are cortical just due to the nature of the injury. So we wanted to see whether this was likely present in the cortex as well. So here we just did immunohistochemical staining of serine racemase with astrocytes and microglia markers, and comparing the perilesional space to the contralateral side, where here we see increased gliosis on the ipsilateral side to injury. And we also saw increased expression of serine racemase as well as its colocalization with astrocyte and microglia cells. So this is suggesting that the mechanisms that we've identified in the hippocampus are likely conserved in the cortex as well. So before I get into the data, just wanted to go over how this is graphed. So here on the left, we have our control samples shown in white, and these are all obtained from commercially available sources. And these are typically people that have died from cardiac arrest and don't have CNS comorbidities. And these are all from cortical regions that match our TBI samples. And then here we have all of our TBI samples shown together, versus to the right, we have them divided based on the area of the tissue. So frontal cortex as well as temporal cortex. And this is just because we wanted to see whether there were any trends that corresponded to the area of injury. This is also graphed based on the time from injury to surgery, where circles represent patients that were admitted to surgery less than a day, and squares represent patients that were admitted to surgery over a day from their injury. And then these are also coded by the severity of injury, where red indicates more severe TBI patients, blue indicates moderate TBI patients, and yellow indicates mild TBI. So when we first looked at serine racemase protein levels, we didn't see any significant differences. However, there does seem to be a trend where the more severely and acutely injured patients have very high expression. However, we wanted to look at this on an mRNA level as well, since after injury protein can be quickly degraded. And so we wanted to look at this from a different way. And here with mRNA, we saw that there was a significant increase in serine racemase expression in the TBI patients as compared to controls. And there seems to be a pattern where the more severely injured as well as acutely injured patients have higher levels of expression. We also looked at expression levels of SLC1A4, which is a transporter for D-serine release. And here we saw significant upregulation of SLC1A4 protein in the TBI samples. And we see a trend towards increased mRNA expression, especially in the acute severely injured patients. We also wanted to look at changes in proteins involved in synaptic pruning. So I looked at expression of C3, which is a complement component that I'll be looking at in our mouse model as well. And we did see a significant upregulation in the TBI patients. And what was really interesting is that we see a significantly higher expression of GPR56 in the human patients. And this seems to be particularly high in the severely and acutely injured patients, suggesting that GPR56 could be a novel receptor involved in synaptic damage in human TBI. Okay, so to conclude everything I've gone through today, in our mouse model, we showed that synaptic damage underlies cognitive dysfunction after injury. And we showed that by genetically and pharmacologically ablating glial D-Serine, we can rescue learning and memory, as well as synaptic deficits. And then importantly, we saw that there's upregulation of serine racemase and related proteins in our human TBI patients, suggesting that what we're seeing in our mouse model is applicable to humans as well. And we've also identified GluN2B signaling pathways, as well as GPR56 expression can be modulated by microglial D-Serine, and showing us evidence as to the mechanism of microglial D-Serine mediated synaptic damage. So with that, I'd like to thank everyone in my lab, especially Dan, my mentor, and thank you all so much for listening, and I'll be happy to take questions.
We go to back to your last data slide on the human. Yeah, that right there. That's really interesting and seems super complicated. It looks like you're pooling the data from less than approximately 1 day to greater than 1 day. Could you tell us a little bit about the rationale for that?
Yeah, so I guess a majority of our patients that we receive do enter surgery in less than a day, but we do have some that have a very wide range as well. So we get some patients that are processed 2 or 4 days after injury, as well as like weeks after their admittance. So we're just trying to see if there's differences based on acute injury or patients that have taken much longer to be admitted to surgery, where we could be losing some of these more acute changes. And I think this is just a grouping that we decided on with our initial sample size, but we're still getting a lot more patients. So as we increase the sample size, we'd like to see if we can have a correlation with time and expression levels. So it's predominantly male. Right now, we only have 2 female patients that are included in this data set. We have received a bit more recently, but we haven't been able to process those yet. Yeah, it's definitely something that we want to look at if we see differences. Thank you. NMDA receptor activation.
So I guess, have you done electrophysiological studies showing like the hippocampal pyramidal neurons are hyperactive that we see sometimes after injuries?
No, we haven't done that. We've done paired pulse facilitation and long-term potentiation. But yeah, we do see deficits in long-term potentiation, but I think not in the paired pulse. That was just in the astrocyte mice though.
Mm.
So I'm going to be repeating this in the microglial mice and seeing, looking at paired pulse facilitation and long-term potentiation mostly.
I like this sex difference in the mice. So Dr. Bramlett, years ago, we were all interested in therapeutic interventions in TBI, and one of the ones that we were studying a lot was therapeutic hypothermia. And we always used male rats and mice, and we found some really nice protection, outstanding protection in male TBI. But when we did females, we saw limited protection with hypothermia in the female mice, which was really interesting. And that's been substantiated in many studies. But if you remove the ovaries 10 days before you did the TBI in the females, they acted just like males in terms of the size of the lesion and the sensitivity to hypothermia. So anyway, this hormonal effect on what you're discussing today might be something to think about as well.
Yeah, definitely. Thank you.
On the female versus males difference in your animal model, because it looks like from obviously the limited number of experiments that you could do, there's basically no phenotype in these female mice, right? At least cognitive phenotype. What about the lesion? Do they have a different type of lesion, smaller lesion to the same injury paradigm that you, could that be a reason why you do not get any synaptic damage?
Yeah, that's a good question. We haven't looked at that specifically in our model. I haven't measured lesion volume, but yeah, I think that's something we can definitely do. And I know a lot of the literature has shown smaller lesion sizes in female mice and also less edema. So I think, yeah, that could definitely be a factor. You might just be more of a mild injury in the females.
Do you see changes in serum racemase and microglia and astrocytes in female mice? Maybe you showed already and I didn't see.
Yeah, we are working with our collaborators to get some images back from that. So far, it looks like we have increased serum racemase in the astrocytes, but not the microglia cells in these mice. But that was preliminary with just a few mice, so we're still waiting on that.